In this video, we're going to talk about lipid digestion, which is going to be different than proteins and carbohydrates because they're not water soluble. So we add to the intestine lipids, they're going to kind of go into a big glob by themselves because they want to avoid the water, unlike proteins and carbohydrates that are water soluble. So first off, when you eat a fatty meal, the first thing that happens is called emulsification. So triglycerides and kind of anything to do with lipids, again, are insoluble in water. So they are going to make a big glob themselves because they want to avoid all the water. So you'll have a big fat globule when you eat. Then what happens is you're going to add bile salts. So you're going to add bile to it from the bile duct, which is leading into the gallbladder, and bile produced by the liver. So emulsification is going to happen when you add bile to the fat, the triglycerides. And what happens is it breaks down this large fat globule, and bile salts have an area that is lipid soluble and water soluble. So it's going to arrange itself where it can surround the fat globules with its lipid soluble side and dissolve in water with its water soluble side. So it can break down all of the large balls of fat or globules of fat into smaller ones, more manageable ones. Then, so that's emulsification. What happens is then pancreatic lipases, so the enzymes that are going to break down lipids, can come along and come to these smaller globules, and there's increased surface area, so it's easier and faster for enzymes to surround all these smaller pieces versus one big piece. If they have one big piece, then it's going to take a really long time to break down, and it wouldn't be in your intestine, it'd go right through. So we need to break it down into smaller pieces. So these pancreatic lipases break down this fat into a monoglyceride and two free fatty acids. So it's going to break it down. Then we get what we call micelles. So as it's breaking it down, we're getting into smaller and smaller globules or little kind of pieces. So now we have bile salts and we have the breakdown. So we have monoglycerides and the fatty acids in these small little pieces. So we're getting towards where we're able to diffuse into the cells. So we get kind of large piece to smaller piece. Enzymes are breaking it down, still mixed with the bile salts so that we can still be water soluble, because even monoglycerides and the free fatty acids are going to be water insoluble. And then remember, diffusion, it's not secondary active like we have with carbohydrates and proteins. Lipids can leave the micelles, so they can leave the bile salts kind of around in the small intestine. They can be reabsorbed and recycled. So we can leave the micelles, leave the bile salts away, and cross the plasma membrane just by simple diffusion. So remember, it's going from high concentration to low concentration. And because of the phospholipid bilayer, so these lipid products can just go right through. They don't need a carrier or transport. So they can go from high concentration to low concentration inside the cell and diffuse that way. So they don't need extra help. So leave the bile salts away because they have water soluble section, and then just diffuse right through. Then once they have diffused inside the cell, so the brush border microvilli, and they've gone inside the cell, you actually convert the monoglycerides and the fatty acid chains back into triglycerides and package them again. So you are having teeny tiny small microscopic globs of fat, and they're made into triglycerides, and you surround them with lipoproteins and lecithin and that can make it water soluble again. Remember, the lipo part is going to be fat, so it's going to be fat soluble, and the proteins part is going to be water soluble. So you're going to get to the point where you can have them dissolved in the cells and can transport them that way. So you go from a micelle and then go into the cell and become a chylomicron. Chylomicrons are then brought to the basolateral side, and instead of, so they are kind of released, exocytosed, so kind of released from the cell, exiting the cell, and they enter not the capillary blood capillaries, but the lymphatic lacteals. So within the villi, we have a lymphatic capillary called a lacteal. So you have blood capillaries, but lymphatic capillaries as well. Remember, they have the big valves, so they can open up wide, and these chylomicrons can be absorbed into these lymphatic lacteals. Then they travel through the lymphatic system, through the thoracic duct going up the small intestine, and then enter the venous side of the bloodstream into the subclavian veins. So they do enter the blood eventually. They just are traveling through the lymphatic system before they do so. Once they travel through the lymphatic system and are in the blood, chylomicrons can be broken further and back down from triglycerides back into free fatty acids and glycerol, and that's done by another enzyme so that they can be used by cells. So those fat products can then be used for whatever the cell needs. Short chain fatty acids, so if they're really small chain fatty acid, they can actually go right into the blood. They don't need to go through the lymphatic system. But most fats are going to be larger and more kind of against water, so more water insoluble, that they're going to travel through the lymphatic system and then go into the bloodstream that way. Just gives them longer to kind of break down and be processed. So first, again, is emulsification. So you have a big fat globule that kind of congeals and comes together. You add bile salts, and they are able to surround the fat and make it into small fat droplets. That increases the surface area for those lipases, the pancreatic lipase, to come in and to break them down. So you break down with pancreatic lipases, you get monoglycerides and free fatty acids, and then you get the micelles. So you've gone from large fat globule to smaller to even smaller. So these are the fatty acids, monoglycerides, and bile salts mixed together in these little tiny droplets. Then they can go to the epithelial cells of the small intestine. So again, they don't need extra help transportation. They can just go into the cell by from high concentration to low concentration. Then once inside their cell, we go back. So the fatty acids and the monoglycerides are then packaged and made into triglycerides again and other kind of any fats, and you mix it with proteins like those lipoproteins to form little tiny microscopic chylomicrons. Chylomicrons are then exocytosed, and then they travel into the lacteal, so the lymphatic system, and then they're carried through the lymph into eventually leading, because lymph leads to the venous side of the cardiovascular system. So just looking at fat digestion again, so we have lingual lipase, doesn't have a lot of help, not as much as going down to pancreatic lipases. So gastric lipase isn't as helpful as well. Most of your fat digestion is going to happen in emulsification with bile salts from the liver, stored by the gallbladder and the pancreatic lipases. Just quickly looking at digestion of nucleic acids. So we have cells that we ingest, and that's going to have DNA and RNA, whether it's in either plant or animal cells. So we have nucleases that are going to break down nucleic acids into those nucleotide monomers. So kind of have those phosphate sugar backbone, and then um, the ACT or G or a U in RNA. So it's going to have those nitrogenous bases with them. The brush border enzymes are going to break down into those nitrogenous bases, the sugars and the phosphates. So it's going to break even the nucleotides down further. So again, this is going to be similar to both protein and carbohydrate digestion. And then we're going to have active transport through carriers into the villi, into the cells of the small intestine. So you can just see we have pancreatic nucleases that are going to break them down. Brush border enzymes are going to break them down even further, and then they're going to absorb that way.